I'm Megan Weber, an extension educator here at the University of Minnesota, focusing on aquatic invasive species. And I'm excited to be welcoming our guest, Dr. Gretchen Hansen, here today to talk about work that she's been doing relating to mercury concentrations in fish in zebra mussel infested lakes. I just have a couple of fast housekeeping items, and then I'm going to turn it over to Gretchen for you. We are recording today's webinar, so you can watch for recording to go up on our YouTube channel. It usually takes a week or two to get through the transcriptions, but it'll be there in case there's anything you missed or there's information that you're hoping to share with somebody else. And we do have mute on right now, but if you do have questions, we're using a tool called Slido to moderate those. So you can go to slido.com and enter the code Mercury, and you'll be able to view the Q&A that way. It also allows you to upvote questions. So once it gets to Q&A time, we can kind of get the most pressing questions answered at the end. I will have the code for that back up once we get to Q&A, but you can enter questions at any point. We just won't be answering them until Gretchen's done with her presentation. And I think that's all I have right now for the housekeeping items. So I'm going to turn off my slide share and I'm going to turn it over to Gretchen to introduce her project and share what she has for us today.
Great, thanks, Megan. Is the audio sounding okay? She's nodding yes. Okay. Thanks everybody for being here and for the opportunity to present. I'm Gretchen Hansen. I'm an associate professor at University of Minnesota in the Department of Fisheries, Wildlife, and Conservation Biology. I'm going to present to you today some work that is the culmination of kind of multiple phases of some projects that I've been working on with collaborators, partners, both students and staff here within the university and partners from the state of Minnesota and beyond, in understanding how zebra mussels are affecting mercury in fish in our lakes. So we've got to make sure the slides are going forward. There they go. Okay. So I think with this crowd, we are familiar with what a zebra mussel is. Zebra mussels are an aquatic invasive species here in North America. They're native to the Caspian Sea region. They were first found in the US in the late 80s, came through the Great Lakes, and then have done some secondary spreading throughout inland lakes in the region. They are aggressive colonizers, and one of their main impacts that has been documented is they increase water clarity and nutrient availability for other organisms, and that's going to be a theme of kind of how they might affect other processes, including mercury, in our lakes. So we'll keep coming back to that one. Just a little side note. This project has focused a lot. It's not just focused on walleye, but kind of started with a focus on walleye. And just to note that zebra mussels are much more likely to be found in walleye lakes in Minnesota than in non-walleye lakes. As of, this is a little slightly out of date, this is from March 2025, but as of March, there were 343 lakes with confirmed zebra mussel infestations in Minnesota. 77% of those are lakes that are managed for walleye. So the infestation rate in lakes that are managed for walleye is much higher than kind of the average infestation rate across the state. So this makes sense. Lakes that are managed for walleye and have walleye fisheries are much more visited than maybe other lakes, just a random lake across the landscape. But just notable that zebra mussels are getting into lakes that are managed for walleye, and so therefore kind of explains some of the focus that my research group has had in understanding zebra mussel impacts on walleye in particular, but also on other fish species as well, as we'll hear about today. So like I said, one of the main, like most documented, most consistent effects of zebra mussels is an increase in water clarity. Zebra mussels are filter feeders, so they are filtering water through their bodies and kind of taking nutrients out of that water column, using them for their own, growth and survival, and then also depositing their waste products on the bottoms of lakes. So they're kind of taking nutrients out of that water column, depositing it on the bottom. That increases water clarity. It also reduces zooplankton biomass. So zooplankton are small animals that live in the water column that are really important fish food, among other things. And so by reducing the nutrients and the algae in the water column, zebra mussels also reduce zooplankton biomass in lakes. As I said, they deposit some of their waste products on the bottom of lakes, so that increases that productivity of lake bottoms and kind of nearshore areas. And all of this can have impacts on fish, although the impacts that have been documented for fish are much more variable than, say, impacts on water quality. So across a range of studies, we see, some declines in fish growth, some declines in fish production, but it's not quite as consistent, really much more variable among lakes and among species than some of these other lower trophic level effects. And so that kind of has inspired, again, some of my lab's work on understanding the impacts of zebra mussels on fish in lakes in Minnesota and what we can learn about, why it's variable or what we can consistently say. So just to kind of summarize the ways that we think zebra mussels could impact fish and walleye and perkids in particular, through their increased water clarity and reduced zooplankton biomass, that could result in a loss of habitat for walleye. So walleye prefer kind of murkier water, so increased water clarity is not necessarily great for them. Also reduced food sources. Larval walleye, juvenile walleye depend on zooplankton in they're kind of when they first hatch. So reduced zooplankton biomass could have a negative impact on the recruitment of walleye because those little fish need that zooplankton before they can transition to fish. It also might kind of indirectly affect other fish species that rely on zooplankton, thereby reducing prey biomass for walleye. And then that increased nearshore production that comes along with zebra mussels could cause walleye to focus more on those nearshore resources and potentially create better conditions for competitors like you say black bass or other centrarchids that might thrive in those environments. And then we might see some impacts on the fishery. So the way that humans are interacting with these species, we might see slower growth of fish if they have less habitat or less prey, lower abundance, potentially reduced stocking success if the habitat is not as good. And then what we're here to talk about today is potentially higher mercury concentrations. So mercury is a human health concern. Methyl mercury is a form of mercury that is a neurotoxin, and it is especially a concern for babies and young children and women of childbearing age. And fish is a major source of mercury for humans worldwide. It is the main source that humans are exposed to mercury. So understanding kind of where mercury is high, which species, and what lake characteristics might influence where mercury is higher is important for human health. But at the same time, fish is a healthy and sustainable food source for all kinds of people. So trying to understand the kind of trade-offs is really important when understanding mercury exposure of humans through eating fish. So in Minnesota and in lots of places, mercury concentrations in fish were generally on a strong decline due to changes in emissions and kind of atmospheric deposition that happened over the past many decades. So you can see, here, where's my cursor? There it is. You can see from this graph from a paper from 2011, mercury concentrations in many species decline from the 70s through the 2000s due to that decrease in deposition. But then in a lot of species kind of experienced some sort of change in trend, and in some places mercury concentrations kind of leveled off. In some places they started to increase. And there were a lot of questions about why that is. And we still don't know all of the reasons why that is. There was a story on NPR just last week or 2 weeks ago about mercury in fish and all of the different factors that influence how much mercury ends up in the fish that we might want to harvest and consume. And one of those was zebra mussel is an invasive species, but there's all kinds of other factors going on too. So this is a really complicated chemical process that we are not going to endeavor to understand all aspects of today. So don't worry, there won't be a quiz about this cycle, but just want to point out that there are a large number of factors influencing how much mercury is in a lake in the first place, how much of that mercury is available to fish to kind of take up into their tissues, and then how fast and how much that accumulates over time. And so there's a lot of variability in the results that we see in a lot of these studies because there's so much going on in terms of what's happening in the watershed, what's happening in the lake, and what's happening with the fish that influences how much mercury ends up in fish. All that said, if we make a list of the things that control mercury in fish tissues, this is an incomplete list because there are many different things. But we know that zebra mussels can influence a lot of these things. We know zebra mussels influence water quality, water chemistry, water clarity. They influence dissolved oxygen at the bottom of lakes. They influence fish growth rates and the food web pathways that fish are getting their prey from. So there's a lot of potential for zebra mussels to interact with these cycles and influence mercury bioaccumulation in fish. And this kind of suite of studies that I'm talking about today, looking at the effects of zebra mussels on mercury in walleye and other fishes, really started when, based on work that came out of the Great Lakes, showing that zebra mussels and quagga mussels, so dreissenid mussels together, caused an increase in mercury bioaccumulation in lake trout in Lake Michigan, due to a culmination of both dissolved oxygen effects and food web effects that I'm going to talk about today. So that kind of kicked off this study. They saw it in the Great Lakes. We were wondering, could the same thing be happening in these smaller inland lakes? So I'm going to talk about food web impacts, and I just wanted to put up this kind of cartoon schematic before we get deep into the food web things, just to try to explain what I'm talking about. So when I talk about a food web, you've probably heard of a food chain of, big fish eat little fish, which eat little bugs, which eat maybe algae. In reality, most of our ecosystems are food webs, so it's not a linear, straight line between one to the next, but there's a large number of species out there that are eating each other, creating this complex web. Walleye are generally at the top of the web, so they are a species that eats other fish primarily, so they are a high trophic level top predator fish. And so we can think of the different ways that invasive species like zebra mussels might influence what walleye eat. So I already told you that when zebra mussels get in, they decrease water clarity, they decrease the abundance of zooplankton, which are these animals that live in the water column. And so that is going to impact both the prey that walleye eat and the larval walleye themselves. So we're kind of cutting off that food source when zebra mussels get in. But at the same time, I told you that zebra mussels increase the production of those nearshore organisms. So when zebra mussels spew out their waste products in the bottoms of lakes, that can increase production of benthic algae there, and invertebrates that live on that, and the fish species that live on that. So it's going to shift potentially where walleye are focusing their predation over into this other pathway. Okay, so that's what we're talking about when we talk about food web impacts. So that's an important piece of what I'm going to talk about. The other piece that zebra mussels can do that influences how much mercury get into walleye relates to their capacity to form dense mats on the bottoms of lakes. So when zebra mussels get to high abundances, you've probably seen them in some of our lakes, they get to these really, really high density clumps on the bottom of lakes. And underneath those mats of zebra mussels, the sediments under there generally are without oxygen. And sediments without oxygen are the sediments where mercury can go from its organic state, which is not a neurotoxin and not available to be taken up by organisms, and be converted into methylmercury through bacterial processes. So in those anoxic sediments, mercury is converted into methylmercury. That is the kind that is taken up by organisms, as represented in this little cartoon by these red dots. It gets into these organisms only when the sediments are anoxic. And then if walleye eat those organisms, which we think that they do because we're concentrating feeding in those areas, then they might have more mercury in them. So this was what was demonstrated in the Great Lakes, and we wanted to test in our studies. Is this going on here? Do we see walleye feeding more in these areas as a result of increased production in the nearshore zones by zebra mussels, and does that lead to enhanced bioaccumulation of mercury in fish? So we did a pretty large comparative study where we went out to a number of lakes. I'll show you the map next, but a number of lakes across the state, and collected all components of the food web, and looked at which species or which areas are walleye eating in. And we also sampled mercury in fish of various life stages to see what was going on there. So it was a pretty intensive field project. And we went to 9 lakes that didn't have zebra mussels in them, so they were our sort of uninvaded reference lakes, and 12 lakes with zebra mussels. This was at the time of study, so some of these have since been invaded by zebra mussels, but at the time the data were collected, we had 12 lakes with and 9 lakes without zebra mussels, kind of spread throughout the middle of the state, where we could compare are walleye eating more in those nearshore zones, and does that result in more mercury in their tissues in lakes that have zebra mussels? So the answer to both those questions was yes. So when we compared fish in lakes that had zebra mussels to lakes without zebra mussels, we saw both walleye and yellow perch across life stages, so adults and age 0, which are the ones that were hatched that year. Both species, both life stages, relied more on those nearshore or littoral resources in lakes that had zebra mussels compared to lakes that didn't. So as expected, that kind of increase in production along the lake bottom resulted in fish focusing their feeding more in those areas. And we observed higher mercury concentrations across species and across life stages in zebra mussel lakes compared to uninvaded lakes. So adult walleye had 72% higher mercury concentrations on average. Adult yellow perch had 157% higher mercury concentrations in zebra mussel lakes. And then we also observed some increases in mercury in those age 0 fish, those really young fish, which really pointed towards kind of ecosystem-wide effects affecting mercury cycling in these places. So you'll note in these figures, and it's going to come out in some other places too, there's a dashed line here on the walleye figure. It's at 0.22 parts per million mercury. So that is a threshold that is sometimes used when creating consumption advisories for fish related to mercury. And it is, in general, it is the concentration that will change consumption advisories from 1 fish per week to 1 fish per month. So it is it's a value, just a baseline value that we use for reference here. So you can see in this case, when we look on average across these lakes, walleye from lakes with zebra mussels, their average concentration was above that 0.22 mark in zebra mussel lakes, but not above that mark in uninvaded lakes. And for all other species and life stages, they are well below that threshold. So these age 0 fish, these really little baby fish, they're not anywhere close to a threshold that's going to trigger a consumption advisory, but we do see increases in mercury in those fish. So mercury accumulates in fish tissue over time. And so older, bigger fish have more mercury than smaller, younger fish. And so the amount of mercury present in a fish and the effect that zebra mussels might have on that accumulation depends on fish length. So when we look at mercury concentrations by length in lakes that have zebra mussels versus those that don't, we see that the average mercury concentration of a 16.5-inch walleye, which was like the average size of an adult walleye in our study, was 1.7% higher in invaded versus uninvaded lakes. And those fish are more than twice as likely to exceed that 0.22 parts per million threshold. We also see that walleye exceed that threshold at a smaller size in lakes that have zebra mussels, so 12.6 inches versus 17.6 inches on average. This is average across all lakes. And for yellow perch, although generally they are well below the 0.22 part per million threshold, we do see large adult yellow perch on average exceeding that threshold at 8.1 inches, but they're really never expected to exceed that threshold in uninvaded lakes. So just another way to kind of summarize those results when taking into account fish size, which also points to some of our ongoing and future work related to, what can we do about this in terms of adjusting maybe harvest or consumption strategies for groups that might be concerned about mercury consumption. So we'll come back to that. Another thing that really came out of this initial study, I should say this was published last year, so this paper is out there for anybody who loves reading scientific papers and wants to get into the nitty-gritty, or you can ask me questions about it. One thing that I do want to emphasize from this study is that we did see pretty high variability among lakes in mercury concentrations. So if we look at adult walleye in particular, we have our study lakes listed here from highest concentration to lowest concentration for lakes with zebra mussels, these kind of orangey pink, salmon colors, and lakes without, the turquoise. And so what maybe jumps out to you here is, yes, on average, lakes with zebra mussels have much higher mercury concentrations, but there are some lakes where mercury concentrations are pretty low and below that 0.22 threshold, even though they're invaded with zebra mussels. And conversely, there are some lakes that don't have zebra mussels that also have pretty high mercury concentrations. So this points to that variability and the numerous factors that influence mercury concentrations in fish that we really couldn't control for in this study. We tried to pick lakes that were generally similar in their characteristics. We tried to, as much as possible, have lakes nearby each other, one that had zebra mussels and one that didn't. But of course, we don't live, like, this is not a controlled experiment. So these lakes were different inherently, and we were just measuring them one time. We just measured mercury and food web characteristics one time in each of these lakes. So it wasn't like we compared, pre-zebra mussel to post-zebra mussel. We were just comparing lakes that had zebra mussels to lakes that didn't. And as much as we tried to control for other factors, we couldn't really do that because we weren't designing an experiment. We were just collecting data on lakes that had already been invaded versus lakes that didn't. And that comes with some caveats, right? So at the end of the day, we're looking at mercury concentrations and how they correlate with zebra mussel invasion, but we're not doing an experiment. And so we've all heard, correlation doesn't equal causation. We did our best to control for things in this case, but at the end of the day, we can't fully discount, other differences among these lakes that might have influenced mercury concentrations. And I always like to throw in a funny correlation just to drive this point home of there are lots of things that are correlated that we do not think are causally connected. And so it's important, my approach to doing science is to look for, multiple lines of evidence when we have to rely on, observational or natural experiments, as is the case in invasions. We want to look for multiple lines of evidence to really kind of build a case for whether this thing is happening or if it's not. We can't go out there and conduct an experiment, controlled experiment, having zebra mussels in one lake and having, one half of a lake and not in one half of a lake. We just have to rely on kind of natural experiments out there. So how can we deal with this? What are other lines of evidence we can use? Well, one thing that we can do is look at changes pre and post invasion. And so this is kind of a next phase of this research. So we have leveraged fish tissue mercury data collected by the state of Minnesota for decades. We gathered those data and we looked for places that had at least one measurement before zebra mussels got into a lake and then at least one measurement after zebra mussels got into a lake. So in this way, we could really look for change and at least start to control for other lake level characteristics that might influence mercury by looking at the same lake over time. Of course, other things could change during that same time period, right? So if a lake was sampled in 2001 and then again in 2021, lots of stuff has changed, maybe not just mercury invasion during that time. So we also looked at uninvaded reference lakes over the same time period to try to understand or build additional lines of evidence to ask whether mercury increased in lakes following the zebra mussel invasion. So in this study, we had 397 lakes, 93 with zebra mussels, just over 300 without, and over 13,000 fish samples. And in this study, we looked at multiple species. We didn't just look at walleye. We were able to do the analysis for these 6 species based on what had been sampled in the statewide data set. Again, we weren't collecting these data experimentally. We're just analyzing what had already been collected, kind of leveraging the monitoring data set to ask these questions. So we had the most data for northern pike and walleye because those are the species that are of most concern when it comes to mercury and human health, but data on other species as well that are kind of lower in the food chain and might provide some additional insight. So we used what's called a BACI or before after control impact design. And this is a design, again, that reflects this isn't a controlled experiment where we can control everything. And we're going to look at changes in mercury, before and after zebra mussel invasion and compare those changes to mercury concentrations over the same time period in lakes that didn't get invaded with zebra mussels. So again, to try to control for what else has changed over the past 20 years that might be influencing mercury. Let's look at some reference lakes to try to understand that. So what does that look like? Here are the figures for northern pike and walleye, the species where we had the most data. So there's a lot going on here. I will try to walk you through it. So what we show here is mercury concentrations before and after. So for lakes that have zebra mussels, again, that kind of pinky salmony color, that's before zebra mussels got in and after zebra mussels got in. So in the case of northern pike, you can see it went from, down here just below 0.28 to up here at about, I don't know, 0.31. So there's an increase. The blue points represent uninvaded lakes over the same time period. So in the case of northern pike, you can see there was a slight increase in mercury across that same time period in the uninvaded lakes, but it wasn't as big of a change as in the invaded lakes. So what we're really interested in is, did the zebra mussel lakes change in a way that was different from the uninvaded lakes? And we do that for multiple species. And this looks really messy and there's lots of lines crossing each other. But like I said, what we're really interested in is the difference between the changes in these two groups. So when we look at that, what we show here is that difference. And if the number is above 0, that means lakes that were invaded by zebra mussels increased more than lakes that weren't invaded by zebra mussels. So we would interpret that as zebra mussels are associated with an increase in mercury in these species. And what we find is that for a number of the species we looked at, there was an increase in mercury associated with zebra mussel invasion. So there was an increase in northern pike, walleye, black crappie, and bluegill, and not a significant increase in largemouth bass or yellow perch. And those estimates were really variable, in part due to low sample size for those species in the data set. We can translate those. A lot of people don't think about, parts per million mercury and what that means. So we can translate those changes into how many meals per month somebody who wanted to stay below a certain consumption threshold might want to consume. So lakes after zebra mussel invasion, you would be recommended to have somewhat fewer meals to stay below a certain threshold across all of these species, but the effect is relatively small. And for species low in the food chain, like bluegill and black crappie, even though you're suggested to maybe consume fewer meals per month if you want to stay below a certain level, you're still able to consume quite a large number of fish meals per month without exceeding thresholds associated with concern for mercury consumption. So we do see increases in mercury associated with zebra mussels across these species in this larger set of lakes, but the change is smaller than what was estimated in the first study on the smaller number of lakes. And again, I just want to pause to note that any discussion of mercury in fish is not intended to be, alarmist or scare anybody away from eating fish. Like anything, there are risks and benefits involved, and in general, fish is a very healthy and sustainable food source by a number of metrics, and indeed, compared to lots of other ways we might get food, has on average a kind of lower environmental impact and a lot of health benefits compared to other forms of protein. So not telling anybody what to do or not do, just pointing out, just always want to remind everybody that this isn't meant to be a big scary, never eat fish kind of message, but just something to consider when we think about impacts of zebra mussels. And that leads to kind of, what can we do about it if a person is concerned about mercury and the impacts of zebra mussels on mercury in fish? Of course, any conversation about impacts of AIS should always lead to an idea of, the best thing we can do is prevent the spread. That is the most effective way to avoid these impacts. So by, following recommendations and laws to clean, drain, and dry and avoid spreading them to any other lakes. In places where zebra mussels do exist and we might be concerned about mercury consumption, it is possible to focus the consumption on maybe smaller fish or lower trophic levels to reduce the amount of mercury consumed while considering the risks and benefits to those approaches. And that leads us to one of our next steps that we will be pursuing in this study of how zebra mussels affect mercury in fish. So one thing that we're doing that we're working on now is working on developing a landscape level predictive model of mercury that leverages data from different species to inform each other. So what does that mean? Right now, the way that data are collected and processed and analyzed to create consumption advisories or just information for people about mercury in their fish, the fish are sampled relatively haphazardly, opportunistically, I would say. So they might sample, just yellow perch from one lake, or, just walleye and northern pike from another. And maybe they haven't looked at all the other species in a lake. So we're working with partners to see if we can develop a statistical model to say, if you know something about lake characteristics and you know something about maybe the mercury concentrations of one species of fish, can that tell you anything about likely mercury concentrations in other species of fish? And at the same time, how does zebra mussel presence kind of influence those predictions? And could this be useful in understanding mercury risk and understanding the drivers of mercury in lakes throughout Minnesota? So that's ongoing right now. And then we just got notified that we have a continuation of this mercury study that will start in January of next year, funded by MASERC and the Environment and Natural Resources Trust Fund, where we are going to dig even deeper into these effects and really try to understand this variability and try to partition out all the different ways that zebra mussels might be affecting how much mercury ends up in fish, to understand the variability in response that we're seeing. So we're going to get in a little bit more mechanistically and look at mercury in kind of all components of the environment and the food web to trace kind of how it gets into fish, and what the major drivers are of that. Is it more about food webs? Is it more about lack of oxygen in the sediments causing more methylation? Is it something to do with fish growth rates, which we haven't talked about much today, but is a driver of fish tissue mercury, how fast they grow. And so if zebra mussels influence fish growth rates, that might be part of the picture too. So we'll look at all those pathways that zebra mussels might affect fish tissue mercury to understand where the risk might be highest and where there might be some potential kind of mitigation strategies where we could kind of insert ourselves into this process and maybe reduce some of the mercury that's getting into fish harvested by humans. And then we're also working with partners to evaluate different fish harvest and consumption strategies that might mediate mercury exposure in places where where fish tissue mercury is increased due to zebra mussels. So we're continuing this work. As always, everything that we do in lake ecology, answering one question leads to 10 more, and it's always complicated and it always depends, and there's always variability across lakes. And so we're trying to really understand that variability when understanding how zebra mussels affect fish tissue mercury in our lakes. So that is all I have. Looks like I'm almost on time. And I guess it's time for questions, and my contact information is here for anybody who has questions. So I think somebody else is going to do the questions.
Yes, sorry, my camera and microphone didn't want to come off, but yes, thanks, Gretchen. This was really interesting to hear, and I'm sure we have questions rolling in here. So I'm gonna take over screen share again for just a little bit. I mentioned that we're using a tool called Slido to manage Q&A. So the code is up for you again, and I also just put it into chat. If you're having troubles using it and you put your question into the Zoom chat feature, don't worry, we'll take it and we'll drop it into Slido there. But you're also able to upvote questions that you see once you're in there. So feel free to find your way in there and drop your questions into chat, into the Slido, and we'll start working our way through those. Gretchen, I'll go ahead. I'll leave the screen up for just a little bit so folks can get the code, and then I'll shut it off so that they don't have to just stare at a QR code the whole time. But the first question that I see is asking if you think some of the variability in mercury concentration among the zebra mussel lakes could be due to a gradient in infestation extent or like the level of establishment.
Yeah, totally. So one limitation that we face all the time when studying zebra mussel impacts is that we don't really have good data on level of establishment, like how dense the populations are. So we really focus on, as I presented, kind of a binary of they're there or they're not there. When we design these studies, we try to choose lakes that have extensive populations. So just based on kind of local knowledge, like, are there a lot of them out there? It's not just, somebody found one once. But yes, especially in the kind of statewide study where we were relying just on infestation based on the infested waters list, that does not distinguish between levels of infestation. So certainly that could be contributing to the variability.
Great, thank you. All right. Next question. Are you able to predict how long it might take for mercury concentrations to be at a level where there's consumption advisories for a species, for any of the species?
How long it might take, like post
Yeah,
zebra
I'm
mussel
assuming that they're
inclusion?
wondering like once zebra mussels come in, like how long does it take for those impacts to start to show up?
Yeah, we don't know that. So again, we've really just looked at, is it invaded or is it not with zebra mussels? We haven't really looked at the time since invasion or if there's any sort of, yeah, how that relationship might look. There's lots of ways it could look. So no, I don't know. And again, I wouldn't, because the variability is so high, I don't think I would venture to say, even if we did have that information, like after X number of years, it's going to be not safe to eat because we are observing all this variability. So what we're trying to do now is understand that variability of like which kinds of lakes are the impacts highest kind of thing, but we don't know that yet.
Okay, thank you. All right. So how do you think that zebra mussels are changing some of the important biogeochemical nutrient dynamics, like the sulfates or dissolved organic carbon, that are controlling mercury uptake?
Yeah, great question. I do not think that zebra mussels, I do not have any information that zebra mussels are affecting sulfate or DOC really. Dissolved organic carbon is not something that zebra mussels really are able to filter out of the water. So uh, it's more that they're impacting the biogeochemical cycles through forming those mats on the bottom of lakes, which can form those anoxic sediments that then promote conditions where mercury will be methylated. So sulfate and dissolved organic carbon are two of the many other factors that influence mercury processing in lakes. Again, that contribute to that variability that we see because it's so more of a lake level or watershed level driver of mercury that is not really affected by zebra mussels.
All right. Okay. So the next question is wondering, so where the mercury is coming from in the first place, and then also wondering if you anticipate that those concentrations will continue to increase. So are we continuing to get more mercury too?
Yes, where is mercury coming from? So, I mean, there are natural sources of mercury just from like weathering of rocks. A lot of the mercury on our landscape that we are concerned about now came from kind of industrial processes like coal plant emissions. But those emissions have been significantly reduced. So technological solutions to that mercury emission from industrial sources have been put in place that led to like really significant reductions in mercury deposition globally. So we have really, as a society, reined that in. But and it's still out there. So mercury that was deposited 40 years ago is still potentially in the watershed or in lakes. And then it's more about these biogeochemical processes about making it available to biota and fish that are kind of independent of what's being deposited at some level. So it's still out there, and there are bacterial processes happening that are making it available. So some of it's buried in the sediments of lakes, some of it's on the watershed and gets washed in, and then various processes make it available to fish, even if it was deposited a while ago.
All right.
Also, I'm not a mercury chemist, so any questions or my answers to questions like that will have to be taken with a grain of salt.
Sure. Next one was wondering about how you get the samples from the fish to ultimately determine what the mercury concentration is. What does that process look like?
how we do it or how
Yeah.
one in general might do it?
It doesn't specify. Maybe the question is both. They're just asking how the samples were collected from the fish. Um,
Yeah, so to sample fish for mercury, you need a tissue sample. So we, um, in our first phase of our study, we went out, we collected fish, using nets, and we collected tissue samples to use for both the food web analysis, so we can analyze their chemistry to understand what they're eating. And we sent them to a partner lab with USGS to be analyzed for mercury in the tissues. And that's the process that's used, by the state of Minnesota, by various tribal natural resource and agencies that test fish for mercury. It really takes like a muscle tissue fillet piece that is chemically analyzed for mercury.
okay. All right. So the next person is wondering if there's a way to find out that if their lake has had mercury testing done on the walleye or other fish present.
Yes, so the state of Minnesota does put out consumption advisories for lakes that show species that have been tested that are above a certain threshold level. From what I can tell, they do not, that does not necessarily tell you if it's been tested and shown to be safe or if it just hasn't been tested. You can just see if it's been tested and it's high. So you would want to check the state of Minnesota consumption advisory website to find
Okay.
that information.
Great. And I see they have a follow-on to this too, wondering if there's a way that people can get their own fish tested too.
Like submit it to a lab and pay for it. I don't know.
Okay. Yep. So for now, relying on whatever we have available publicly, most likely.
Yes.
All right. Okay. Next question was wondering if there's any recent cases documented of increased mercury in humans that consume fish in the area studied.
Hmm. There have been studies showing higher mercury concentrations in humans in certain parts of Minnesota. I was not a part of those, and I don't remember how recent they are, but in the past couple decades. Um, yeah, I probably shouldn't speak too much about those because it's outside of my area of expertise.
Sure. Yes. We'll try to stick to fish as much as we can. Okay. Let's see. I see a question here, kind of coming back to that first question about what level of infestation might mean. And this person is using Lake Mille Lacs as an example, but would there be a way to use mussel density over time for lakes where we have that kind of data to help answer that question looking backwards?
Yeah, that is something that could be possible. We just don't really have, Mille Lacs is a pretty special case. We don't have that kind of info um, from a lot of lakes, but yeah, you could look at density over time and how that relates to a variety of impacts, including mercury.
Okay. Great. Okay. And I see a question here wondering if attendees will get an email when this is available on YouTube. So I can take that question and say yes. Yeah, you'll get an email once it's available. We send it out to everyone who registered. So you should get a notice that that's available then. Okay. Um, let's see. I see a question here. Wondering, and I think you might have answered this one. I'm not sure if this one at the same time, but just in case there's any extra nuance to it, they're wondering if you could go over again what the sources of methyl mercury are.
Methyl mercury. Yeah. So methyl mercury is, well, again, I'm not a mercury biogeochemist, so I'm going to explain to the best of my ability as a fish ecologist. Methylmercury is the form of mercury that is toxic to humans and available to be taken up by organisms. And it is formed by bacterial action. primarily in anoxic environments. So there has to be like the inorganic form of mercury there, like some mercury is there, and then the conditions have to be right for that bacterial process to convert it to methylmercury. And generally that is in anoxic environments. So it happens a lot of in wetlands and anoxic sediments in lakes. And so there is a biochemical process through which the mercury is methylated by anoxic bacteria that we could pursue understanding the details of that chemistry in a different webinar probably. But it's really bacterial action in anoxic environments.
Okay, great. Okay, I'm going to shut my slide share off because I think folks have this now, and that way we can maybe have a slightly different view. Okay. I see someone who's wondering about if you're able to share the publication from your study and then the Michigan study that you mentioned. And I'd say if you don't have the links available, easy to grab now. If you send them to us, we can also send them out in a follow-up email.
Yeah, certainly yes. Do you want me to do it right now or?
We can keep working our way through questions and maybe we'll
Yeah.
just say watch in that follow-up email and we'll be sure to get them from Gretchen and then can have that information for you there.
Yeah, and certainly the one that my lab led from last year is open access and is linked on the MASERC page and all that. But I could dig up the other one too.
Okay, great. Okay. I see someone asking about a list. I'm wondering if there's a list of infested lakes, and I'm not positive if they mean just zebra mussel lakes here or if this also relates to mercury or not. So maybe I'll just leave it at that and see if you have some thoughts.
A list of lakes that have zebra mussels?
I think that might be what they're asking, but for whoever asked this question about if there's a complete list of infested lakes, feel free to add a clarifying point if I'm misunderstanding your question.
Yeah, I mean, the state of Minnesota maintains an infested waters list that's updated fairly regularly. So yes.
Great. All right. Yeah. So not knowing where this person is joining from, if you're in Minnesota, it should be pretty easy to find from Department of Natural Resources. If you're outside of Minnesota, you might need a different data source.
Oh yeah, I guess I was thinking Minnesota specifically.
Yes.
Yeah, here, I don't know, can they see what I put in the chat? So I just put in the chat a link to the article and also the infested waters list.
Great. Thank you. Yeah. And I think Michelle is in the background there. So if it's not available to participants, she can make sure it gets available to them. All right. Next question I see is says that they've seen some studies show that selenium can buffer mercury, and they're wondering if you've looked at content in this area or if that's another potential follow-up strategy as part of your work.
Yeah, so I know that's like both in terms of human health, like people eat selenium if they've been exposed to a lot of mercury. And there was a study that came out maybe last year looking at like in terms of lake chemistry, the interaction between selenium and mercury and methylmercury in fish. We have not looked into that, but it is something that we are considering, like especially because this new study came out showing that it did have a kind of buffering impact. So we don't have a lot of data statewide on selenium in lakes, but might be something to pursue in the future.
Sure. All right. Another question about, potential future directions. Wondering if either you've already looked at synergistic effects between zebra mussels and other lake characteristics on the increases in mercury, or if not, do you have thoughts on what other variables might be important?
Yeah, I mean, that's part of what, so with this predictive model that we're building, a big part of that is testing the role of other lake characteristics. So we know for sure that watershed characteristics, particularly like watershed size relative to lake size. So lakes with big watersheds relative to the lake size tend to have more mercury. We know wetlands and organic carbon and sulfate concentrations, as we just talked about, also influence how much mercury is in lakes and how much methylmercury is available. So we are incorporating those characteristics into our predictive model to try to tease apart how much zebra mussels might explain, once those other things have been accounted for.
Great. All right. I see just a couple of notes. It looks like someone had some other information here about following up on where different contaminants, or sorry, where you can find information about tested fish. So they said you can contact Minnesota Department of Health to find guidelines in water bodies that have been tested. That Department of Natural Resources has information on the Lake Finder page for fish that were tested for the different contaminants, and then a note that Great Lakes Indian Fish and Wildlife Commission and some of the tribal nations also have consumption guidelines for tribal nation members.
Yes, thank you.
Okay. Okay, one more question here. So this person says a shift from pelagic to littoral anoxic food sources increased association with areas of high methylmercury. How else did the observed or tested fish species in zebra mussel versus non-zebra mussel lakes differ? For example, age classes, habitat use, or internal loading differences?
I'm sorry, because I don't have that in front of me. I'm going to need you to say it again.
Yep,
I'm a
here.
visual learner.
I'll also drop this into chat
Okay.
for you quick too. I am the same way. One sec. My Zoom wants to cooperate. Okay. So if a shift from pelagic to littoral anoxic food sources increased association with areas with high methylmercury, how else did observed or tested fish species in zebra mussel versus non-zebra mussel lakes differ? For example, age classes, habitat use, or internal loading differences?
Yes. Okay. We don't know for a lot of those things. We are particularly interested in age and growth differences. And so that is something that we're looking at in this next round of study. Unfortunately, in the statewide, like historic Mercury database, we don't have fish age associated with those fish. We just have fish length. So we account for length, fish size and all these things, but you can have a fish of the same size that is different, a different age, and that's going to impact how much mercury is in it. So that is an area of interest for this next round of study, is looking at how age and growth is both affected by zebra mussels and how that might translate to differences in mercury accumulation. Habitat use, we don't have information on individual fish and where they're hanging out. The food web study that we did is sort of a proxy for habitat use, or at least, prey utilization, that we can say, these are feeding more inshore versus offshore. So that is sort of a proxy for habitat use, but we don't really know, like, where are they spending all their time, if that's what we mean by habitat use. And then I'm not entirely sure what internal loading differences is referring to.
Okay, great. All right. I see a question from someone who is on a lake with zebra mussels, and it sounds like they tend to crush up the mussels and maybe put them off their dock or something, it sounds like, and that they notice that the sunfish really like to eat them, and they're wondering if you can see any like potential negative impacts with that kind of a process. I'm sure at least as it relates to this study.
Um, Not that come to mind right now, no. Seems like a nice way to feed the sunnies.
Yeah. All right, let's see. And then I see a question, maybe more about zebra mussels here, but wondering, so they're asking about zebra mussels and increasing water clarity. So they're wondering if that means you might see differences in Secchi depth. The example they gave is like maybe it would be a change from 10 feet to 12 feet. Is that what you're describing?
Yes, or even more.
Sure.
Yeah, even bigger differences are common.
Great. All right. I think we've made it through the questions here. I think all I see left are just a couple of comments. So I'm going to start up screen share again, and we can go over our final housekeeping items, and that should have us wrapped up just in time here. So I'll keep my eye out in case any last minute questions pop in too. Um, but first of all, thank you, Gretchen, for joining us. It was really great to hear the results of this work and see some of that helpful information. Um, I mentioned that we are recording and that it will be available. So you should be able to see this on YouTube. Again, give us like a week or two to do transcriptions so it can be nice and closed captioned. But we'll send out a notice to everyone once that becomes available. And you will see a survey that'll either pop up as you leave or you'll get an email. We really appreciate if you can take some time to fill out that survey. I did put up contact information for you, so if you have additional questions about the study or the content of Gretchen's presentation, her email is showing on the screen now. And then if you have questions for us as webinar hosts in terms of, other webinars, recordings, our program, our email is aisdetectors@umn.edu. Um, and I'm going to just double check one more time before we sign off that all of our questions got answered. I don't see anything new in the chat. So I'm going to say thanks again, Gretchen. I really appreciate you sharing your time with us. I know it was a puzzle getting this scheduled, but I'm really glad we were able to find a time for you to be able to join us. And thanks again to everyone who tuned in to listen to the webinar this week or this month. Next month, we have one more webinar. It's a Starry Stonewort webinar, and we're welcoming Raining White and Steve Smith to talk about Starry Stonewort management around wild rice. So we hope that we'll see you at that webinar as well. Thanks for joining us.
Thanks, everyone.